This is part 2 to lecture 19. We're going to talk now about the spontaneous mutations that can occur from replication error and what we'll call naturally occurring base or nucleotide modification. It's important to remember that no cellular process happens perfectly. There are always errors that occur. And those errors during mutation events can be due to slipping of DNA polymerase, or they can be due to mis or incorrect base pairing, which can occur as a result of what is called tautomeric shift and or base damage. Let's talk about these in some detail now. One of the ways that errors can be introduced is the slippage of DNA polymerase during routine DNA synthesis, which happens whenever you copy the chromosome in anticipation of cell division. And where this is usually observed is in areas where you have repeated patterns of bases, for example, CAG. So you have a CAG, 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 CAG. In this figure, part A, that's represented by having a long stretch of the same color here. Okay? And what will happen is the DNA polymerase seems to get somewhat chemically confused as it's traveling over this, and it will copy, it will make too many, a stretch that is too long on the new strand. So the new strand is shown on the top, and the template is below, and DNA polymerase essentially got carried away and it made too many copies of this CAG repeat. And the result of that is that a bubble forms. And when the bubble forms, it distorts the double helix that's resulting from the process of DNA synthesis proceeding in a normal semi-conservative fashion. And the DNA polymerase is able to detect a distortion. In nature, the nucleotide bases can in fact occur in different forms that differ in the positioning of a single proton. And that can result in alternate base pairing arrangements. So the standard base pairing arrangements are shown in the top of this figure, okay, up here. So in the normal configuration in DNA, what happens most of the time is that thymine base pairs with adenine, as shown here, on the top left, and cytosine base pairs with guanine, as shown on the top right. So let's take a look at the thymine on the left-hand side. This thymine is in the most common naturally occurring configuration. It's called the keto form. And in the keto form, note that there is a proton here, okay, and not here. Now in nature, thymine will spontaneously shift back and forth between the keto and the enol form, and the enol form is shown in the bottom left. Note that in the enol form, there's a proton that is covalently bonded, a hydrogen covalently bonded to this oxygen now, and it used to be there. So we had the migration of that proton from place to place, okay, and that will just occur in nature as a result of electron movement. And so when thymine is in the enol form, it favors base pairing with the standard form of guanine, the keto form shown on the right. So now we have a TG base pairing scenario, and that's a little unusual. That's non-standard or anomalous base pairing. Similarly, when cytosine is found in the somewhat unusual, but still naturally occurring, amino form, it can base pair not with guanine, but with adenine, as shown on the right. The adenine is shown in the amino form. It's also a somewhat rare form. Sorry, no, it's the standard form. It's correct, pardon me there. But anyway, these alternative forms, or tautomers, as they're more correctly known, can result in non-standard base pairing. Because they occur naturally, meaning from time to time the common form will shift to the rare form, then during that moment when it's shifted to the rare form, if synthesis is occurring, then you can have non-standard base pairing. And that will persist into the next round of DNA division. It will be a mutation. So let's just look at a cartoon that sort of depicts what I'm saying about a mutation becoming fixed or persisting so that it becomes a true mutation. If you look at this figure, on the left, we have the scenario where there's no tautomeric shift. All of the bases are staying in their most common tautomeric form. And on the right, we're seeing a situation where this adenine is shifting. It's making a tautomeric shift from the common to the rare form, from the into the amino form. So if we follow the right-hand side, this tautomeric shift into the amino form has resulted in non-standard base pairing here. That's an AC base pairing arrangement. It's non-standard. That C has been incorporated in this strand, and if that strand then persists and goes through another round of cell division, the C is there, and it remains there. And after a full round of division, it results in a mutation. In this case, it's a transition mutation. We've defined that in the previous lecture. On the right-hand side, the tautomeric form of the adenine shown shifted back to the regular form, and we actually returned to the original sequence shown back up here on the top. But it doesn't matter, because in all of the cells that will contain that DNA on the left, that's now mutated DNA. If that occurred in a germ cell, then that's now a mutation that will be passed on to offspring. If that occurred in a chromosome found in a somatic cell, you now have a somatic cell mutation, and who knows what the impact of that will be on the life of this particular organism. Now, another thing that can happen to the nucleotides, the bases in our nucleotides, just as a result of sort of routine cell metabolism and naturally occurring processes, is that you can have damage occurring. And the damage can result in deamination or depurination. And when that occurs, you can also have mispairing or altered base pairing arrangements. On the top part of this figure, we have cytosine, okay? And the amino group is highlighted in red. Now, if that amino group is lost, then the base that is left over is uracil. Remember, we see uracil normally in RNA nucleotides, but not that commonly in DNA. It's a nucleotide that is typically associated with RNA, but it can occur. And it can occur as a result of the deamination of cytosine. And when that happens, if that strand becomes the template during DNA synthesis, which it inevitably will at some point in time, uracil will base pair with adenine. And that's different, so we go from having a CG base pairing to having a U-A base pairing. And that means that we've got a one nucleotide change at that position. Because when that goes on, you're going to have TA base pairing that's going to persist. Okay. Similarly, down on the bottom, we've got adenine shown on the left. Here is the amino group of adenine, and if it is lost through a deamination process, the result is a nucleotide, or a base rather, called hypoxanthine, and it's capable of base pairing with cytosine. So instead of having an AT base pair, you would now end up having a CG base pair at that position. So it's a point mutation. Rather than deamination, you can also have the situation where you lose the entire nitrogenous base. And this happens most commonly in purines. And it occurs when the glycosidic bond that holds the nitrogenous base onto the rest of the nucleotide. Okay, so here's the base here. That's the glycosidic bond that I'm talking about. It's a glycosidic covalent bond that's holding any nucleotide's base onto the 5-carbon sugar that is part of the entire nucleotide structure shown here. If you knock that base off, you're left with essentially a headless base, or a depurinated base. And it's usually, this only happens in purines, adenines, and guanines. It can happen to cytosines and thymines, but it's much less common. And it leaves behind, rather than calling it a headless base, it's usually called an apurinic site. And if that is not repaired, and there are enzymes that can do repair, but if that is not repaired, then that headless base is able to base pair with anything. So it'll base pair randomly with whatever nucleotide happens to be around during DNA synthesis, and the result will most likely be an error in at that position on the DNA strand that is forming. Normal cell processes, such as oxygen metabolism, as well as exposure to radiation, can produce free radical chemical species. Those free radical chemical species can damage bases or even induce DNA strand breaks if extreme. Damaged bases are typically repaired using something called nucleotide excision repair, which relies on a set of DNA repair enzymes called glycosylases. But it is important to note that they're not always repaired, and those damaged bases can participate in altered base pairing arrangements that give rise to mutation after a round of DNA synthesis. Let's look at some pictures of the kinds of damaged bases that can often or commonly occur in nature. This image is taken from the website with the address indicated here, and it's the website of Dr. Susan Wallace, who is a microbiologist and molecular biologist at the University of Vermont, and she studies DNA repair enzymes. And shown here are a number of damaged or altered nucleotide bases. And those that are shown in darker or bolder font and lettering are those that result from oxidative damage. So there's a significant number of these that can occur in nature. And what Dr. Wallace studies is how you can have DNA repair enzymes that are able to deal with all of these different types of damaged bases, what their substrate specificity is, and what the evolutionary implications are of those questions. And she found that these are actually pretty ancient repair enzymes that can deal with this type of oxidative damage. Now with that, this concludes the second of this 4-part lecture series. So please go on and listen to the third part.